السلام عليكم ورحمة الله وبركاته. قال أعوذ بالله من الشيطان الرجيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. We're here inshallah ta'ala in the second last lab for class 2, BIOA02. May Allah subhanahu wa ta'ala make it easy for them and all of us. Ameen ya Rabb al-Alameen. Stay tuned. Barakallahu feekum. Looking forward to seeing you soon. Just remember to bear in mind that your lab is it's the one lab. Yeah, they use this for one lab. I think enough for that. It's you may also use gloves during this after school. So just realize from the separate notes. If you guys are wondering, have like questions you want to reference as we're nearing the end of our practical together, let me know. I'd be happy to help you as you pursue, let's say, med school, grad school, wherever you may plan to be. I just wanted to share that. But whenever you're ready, you could begin. You can even begin the pre-lab in about 5-ish minutes or 2 to 3 minutes at 10:10. If you haven't started. How much time would you need to complete the pre-lab if I could get a use of hands of 5 minutes? Is that okay? Yeah, 5 minutes. Okay. Anyone need more than 5 minutes, by the way? Just for context. So we'll wait another 6 minutes just in case. Remember, as we do pre-lab quizzes, to make sure that we're not speaking with one another, very important. I don't want that to impact your grades, so just bear that in mind. If anyone needs also a calculator, please let me know. Yes. Okay. I saw you putting your computer inside. If you want, you can keep it on your bench. No. Okay. Just want to get a confirmation. Are we almost done the pre-lab quiz? Yeah. Do we need another minute or two? A minute? Okay. Okay. You can wait a minute or so. For those that are done, they could also put on gloves. They could get themselves ready as you'll be touching live organisms, plants and invertebrate included. Okay. Let me just confirm. Is everyone done the pre-lab quiz? If you're not, please let me know. I just want to make sure that way those that are still doing it are able to concentrate. Joshua, you're done. Just to confirm, yeah. Everyone done the pre-lab quiz? Okay. So maybe we can get started. Just give me one moment. Okay. So let's get started now. Um, we're at 10:19. So I'm gonna try to go through the intro in due time. You guys did the pre-lab quiz. Now that's exercise one. Exercise four is your post-lab assignment. Do 24 hours from 1 p.m. or 24 hours from today. And we'll pretty much go in reverse order. So go to exercise five, lab five, and that's starting on page 74 of your lab manual. And then I'll talk about that, and hopefully we'll go through instructions, try to be done it in around 25 minutes with instructions, so probably around 10:45. And then examination for about 20-ish minutes should be okay. So that will give you about until 11:05, and then the remainder of the practical will focus on terrestrial isopods. So that portion will likely be 40-ish minutes. It says 50 minutes, that's also okay. And then the remainder for cleanup and even for stats analysis as shown here. So the idea is that you guys can actually perform the data analysis herein within the practical, which will make it easier for your lab 5 assignment. So bear that in mind. Try to finish the analysis, go through the slides with me as we go further into the lab. And inshallah, you'll finish ace your lab 5 assignment. Any questions before I move further? Uh, yeah, was there someone beside you? I want to also make sure, how many people do we have here? There was one person on that side, right? No? Hmm. So technically we need 6 groups for today. So we'll see how to adjust things in due time. Actually, let me just confirm. Anyone from any groups around here notice someone who came in but just left momentarily? Because I thought there was one more person on this side. No one beside you, though? No? Okay. And likewise, no one on this side. Okay. Okay, so let's go further then. Talking about population growth, this is exercise 5. And hopefully this will give you some history or like some memories of lab 1 going into lab 2, because we're actually preparing for our final lab through this exercise. And so pretty much if you look at pages 75 and 76, you'll see that pretty much we're trying to, within groups like before in 6 groups in total, each group will be focusing on a specific condition. And the aim is to be able to look at the effect, observe the effect of different or changes in road salt concentration, i.e. NaCl, on lemna growth. So think of that like your winter wheat assay across drought regions. So hopefully, like you'll see different things connect interestingly. In this case, our analysis is using pi-squared models, but nonetheless, I want you to consider the following. If you look at page 75, you'll see a formula. This is the formula you often see in Chem as well. C1 times B1 is C2 times B2. It shows you the relationship between dilution, often in chemistry, titration, but in this case, we'll be focusing on these two because you want to be able to understand and actually accurately measure the concentration or the volume that you need to have an appropriate concentration. And what I mean by that is if you look at page 76, you'll notice that you have the mention of your source as V1, volume. So 1 refers to the source, right? So just bear that in mind. And then 2 refers to the final volume, final solution. And that would include, actually, I won't talk about that as yet, but the idea is that you want to be able to solve V2, which is right here. And how do you do that? Actually, I think it says V, doesn't say V2. Okay, it says, let me just make a small edit. V1 is done. The idea is very similar. So for context, you're starting off with those larger beakers or vials. And so, by the way, folks, you'll be coming to, depending on your group, a specific drug with a specific concentration. So we have, for example, stock concentrations that are transparent and artificial pond water, which you're going to use to dilute your concentration of your stock appropriately. So think of it like this. Your source is going to be of NaCl at a specific concentration, okay? Which is going to be shown as C1. And then V1 is what you're calculating, and we'll talk about that in due time. V2 is actually going to be incorporating your NaCl, liquid, diluted version or the dilution of your NaCl, plus your artificial pond water, short form for AAP, just for simplicity here. The idea is that if you use this formula, you're going to get, and you just essentially solve for V1, you get C2 times V2 divided by V1. And the idea is that so let's look at the manual. Let's see what volumes or parameters we have already at play. You know that V2, your final concentration, independent of your final volume, independent of your condition, is going to be 200 mils, right? So we know that this is 200 mils. And you want to bear in mind the units, 200 mils is 0.2 liter, right? And then you have concentration, which we already know based on the specific condition. And then you have your stock as well, which is also known. So let's do an example to make it easier. By the way, is anyone confused as I go through a bit of math? Everyone paying attention? Everyone's paying attention? Yep. I can see. So just bear in mind, because I want you to all be able to do this easily. Let's say your condition 2. What's the initial stock concentration of condition? Can anyone share? Actually, let's say, so if you look at page 76, experimental treatment 2, what is the concentration that we're working with, the stock concentration? That's 1.25. That will be the final. Have a look, folks, at column 4. Sorry? Yes, but in the stock, how much do you have? Yes, yes. Did you say 1.25 before? Yeah, I said I was doing the first category. I see. And actually, that's something that we may get confused with, folks, so just bear in mind when paying attention. Condition 2, if you look at the initial, so everyone's paying attention? Let's go with the initial concentration, which we see at 12.5. 12.5 milligrams per mil, we say grams per mil, right? Because 1,000 milligrams is 1 gram, 1,000 milliliters equals 1 milliliter, 1 liter. So that's 1,000 divided by 1,000. Essentially, it's the same for milligrams divided by milliliters. Same applies to other units. But your final solution concentration of NaCl is going to be 1.25. Right? So if you do the math, based on what we see here, 1.25 times 0.2 divided by 12.5, what do you expect to see as your final volume, or your initial volume, essentially, for this equation? Does anyone know? You could use a calculator, you can do mental math. 1.25 divided by 12.5 is what? Does anyone know? 0.1. And I want you to think about that because it's going to be very important. So 0.1 times 0.2 is what? Anyone know? Yeah, you guys. 20 milliliters. So that's going to be your answer for experimental treatment 2, meaning that you start off with 20 ml in your beaker. And I know we have a larger one, but you technically can use a smaller one. This is just purely shown for illustration. If you have NaCl, and we have it at 12.5 grams per liter. If you go up all the way to your final volume of 200 milliliters, then you should expect to see a final concentration of 1.25 grams per liter. And this is important because we want to be able to control, of course, right? The correct volume and the correct concentration that you're using appropriately for your analysis, but also to make sure that, you're doing things stepwise. For what I mean is that you'll see the different conditions go up numerically twofold. For example, the stock concentrations from experimental 1 goes from 6.25 to times 2, 12.5 times 2 to 25 times 2 to 50, and then 50 times 2 is 100. So it goes up by multiples of 2 from experimental treatment 1 to 2 to 3 to 4 and 5. Yet, something to note is that the final concentrations are all 10 times less or 10 times lower. And what that means is that if your concentration is 10 times lower, right? So you divide it by 10. Accordingly, you should have your volume. Well, actually, you can do the inverse. Inverse is better so you guys understand. The inverse of you doing this formula once again, very simple. It just reflects that you now have 10 times of a smaller volume. So that means, like as we showed earlier, right, you're starting off with 20 mils to reach 200 mils, and that leads to a 10-fold decrease in concentration. So pretty much as you look now, and maybe I'll ask you, if you look at experimental treatment 1, you have the stock at 6.25 and the final concentration at 0.625. So because of that ratio, you know it's 10 times less in concentration. What do you expect then should be your initial volume of NaCl? 20 milliliters, right? Does everyone understand that, by the way? Who doesn't understand that and wants me to repeat? I don't mind, but I want to make sure you all understand this. We'll do another exercise very shortly. So if you see the same for experimental treatment 3, you find a 10 times or 10-fold decrease in concentration from your initial to your final. What do you expect then is your initial volume if your final is 200 milliliters? Anyone know? So if you're decreasing by 10 times less, and your final volume is 200 mils, what should be your initial volume to start off with? I heard Bishop say 20. Joshua 20. Who thinks it's 20? Raise your hand. Who thinks it's 10? Who thinks it's 50? So it's 20. And I just want to make sure you guys understand that because the same ratio applies for all conditions. Meaning for your column 3, you should consistently, independent of your group, only put 20 mils into the small glass cylinder here. So bear that in mind, folks. You put 20 mils of your specific concentration, and you'll have cups of 3 reps where you'll be able to replicate this accordingly. But the other point is the volume, and that's shown in column 5. What's the volume of artificial pond water? Remember, the total is 200 mils, BT for short. And BT includes the volume of your artificial pond water plus 20 mils, right? Which is your stock. Concentration of your stock, which we showed was V1 or V NaCl. So if you know that, if we just do the math, this is 200 mils, this is consistently 20, and this is consistently 20, that's consistently 200. What's the volume of artificial pond water that you'll be adding across your condition? I want to hear it. Everyone say it. Say 180 milliliters? What do you think? 180? Salah, what do you believe? I think I should ask everyone. Charlotte, how much are you adding? Sorry? 180. Everyone understand this because I want to make sure that you understand these concepts that you learn also in organic chem. Make sure that you read general chem. You want to be able to understand to do quick math as effectively as possible. So you can essentially fill in now the respective volumes that you should now know for columns 3 and 5 for page 76. Now, the reason I asked about the groups, by the way, any questions? Any questions, folks? No questions? Let's pay attention, folks. You'll have time to talk soon, but I just want to make sure you're all paying attention. Bear in mind now, except for the control where you're not adding any stock, you'll have 200 mils directly added for artificial pond water. Now, you'll be having cups accordingly, 3 cups per condition. And the reason I asked about groups is that we probably will need to split up a bit more. I just need to think. Um, okay, so is it okay if 2 members, so 1 member per group, can move to this side to join random? Reason being is so that way we could make things equal, because technically there's 9 people here, so we can have 3 groups of 3. And that way we consistently have groups of 3 that we could work with for today's practical. And don't worry, we're all a team, one big family, so that should be okay, I hope. Do you guys mind whoever could do so? Yes? Is that okay? I really appreciate it, Joshua. Anyone else? Who wants to go? Who can go? I don't want to pick anyone because that may not sound fair. Choose one other member from group 1A, 1B. Another idea I'm going to pitch is that if you're comfortable, the 2 of you, and need help, I'll help you guys out. I feel like you guys may want to stick together, but if you guys want, that'll also be beneficial, helping out other group members. Both, I do want to share as well, these are going to be your conditions. So as we recall, right, we go 1, 2, 3, 4, 5, 6. So accordingly, you're going to be doing using specific concentration of your stock. So please bear in mind that you're looking at the right stock for the specific concentration of interest. I'm actually gonna have to go use the washroom, but briefly before I do, um, bear in mind that you have the concentrations also in your notebook. You have group 6, actually, so this is perfect. You guys can be the control, the 2 of you. Group 1 could be, actually, you'll see it from page 76, but you're going up essentially in treatment concentrations. I want you to bear in mind once again, um, that as you see here, you have your concentration of your stock of your artificial pond water. Make sure you're adding 20 mils only for each of your reps. Each group is doing 3, and your goal is essentially to do this as efficiently as possible. Um, Yeah, oh, any questions? I know, so I went through it pretty briefly, but the idea is that you're adding in 180 mils of artificial pond water, use the large glass cylinder. For the smaller one, use it for your stock at 20 mils. And you're going to do that, repeat it for 3 different cups, as you can see, and you're going to label them accordingly for the specific condition or concentration with the practical number and your replicate, as well as candidates, so that we keep track of it. 2 weeks. Any final questions for you? No questions? Are you ready to go? Before you move further, just make sure that you orient yourself with your group to the correct concentration. Remember that it goes this way, it's not from 1 all the way to 5 down that way. And then when you're done, I should let you do that. So you can just allow that. Get ready, folks. All the best. I'll be back. Yeah. What you just mark? Yeah. No, we're 3. Yeah.
Oh, okay.
This one, yeah. No, it's
Back to 40. Yeah. Experiment control through 3. What's the computation? Yeah. Alright, I will add the stock, you will add pond water.
You're 40.
I'm adding 20, you're adding 180. Can't add it though. That's like against the rules. Are you close? Yeah, that's close enough. You have 180 million. The difference is like.
No, that's unfair. That's unfair.
Yeah, that's fine. Oh, wow. Dude.
But you point. I'm not. Yeah. So can I go forward? And then
Sure, okay. You start adding plants.
Also add the replicate, so like ABC.
Take your best judgment. I want to watch it. How much overburden? Okay, so you have it. That makes sense. Yeah. All right. I think it's typically class with I.
yeah.
Full healthy plants. To 3 from. Going well. Yeah, that's some plants.
I can
I don't want to spill it. Well, okay, you do it. That's okay.
Oh, yeah.
Like it's not.
So you could tap the middle
like the top of the glass, and then kind of just gently like shake it.
Okay. Oh, yeah, okay. Thank you. You just did that. Well, yeah. Four. Five. Can I just take one out? I just take out that. Yeah. So, like, is this? Yeah. Four. Oh. Oh. No, that looks like solid. I need one more. What looks like two. Okay.
Yeah.
We'll see. Can it separate? Well, let me see how many I
Do you guys have any questions?
I This is okay to do that. Yeah. Wait, let me see. Sorry.
But do you need help like calculating?
Yes, sir.
They have some
2 liters. Can't figure out why it's 12.5 here.
No, they have their one.
12.5 of constant. Maybe concentration.
Like this. 5.
No, and the AC one.
Bars? Yeah, that's.
Yeah.
Okay, okay, yeah.
That's your.
Yeah.
Take care of this, I'll take care of the carpet.
Okay, so it will be 6.
Well, I think this is done.
Yeah. Okay.
Terrestrial export.
Oh, we need lids. I just had to move most of them.
Okay.
So students, just to follow up, just to confirm, how many of you have added in the, or actually how many of you are in midst of adding in the Lemna? You already done? Okay. So I believe the helpful lab tech gives some tips. Did she share anything?
Yeah.
Okay. So briefly, for those that are still trying to do so, I recommend having a member of your group also hold one of the glass cylinders, because especially the one that's larger, you don't want it to tip over. Now, when it comes to the lemna, make sure that for the 4 of them, that they're healthy and they have 3 dahlias or like miniature leaves. So make sure you guys have that. Visualize and make sure that you do see this. And I want to cross-check it for all groups. So that way, you know, down the road, we'll be able to see and hopefully observe reliably.
Yeah, the one does. They're not this. These all look pretty good. Yeah. No, 4/2 That was very good. None of them have really. Look at them all, they're all 2 leaves. How many more do you need? Yeah. And then you follow this. Yeah, mine was like. Yeah. Plenty was. Sometimes sometimes they flutter around to the end. You could really, hopefully, that's very yeah. Yeah. I want to. We'll wait for another few minutes before we move further for our next set of practical exercises.
Really good.
Can't see it.
Just as a reminder, not to label all of this, it's just on the slide. You want it to be done. It's on the slide.
Yeah.
Really?
All right.
I thought this was part of life. Yeah.
Can you guys bring a marker to label the wrap? You need to label.
Yeah.
Oh, I'm so sad. Another one?
You will not get. Why you laughing? This guy went. He tried pouring the whole thing into that.
I will be did it.
Oh, yeah.
So this is, by the way, to be consistent, folks. You have 4 different dahlias across, so each graph is going to supposedly have 4 dahlias, sorry, 4 lemnas with 3 dahlias each. We want to be consistent across not only your own maps for the your specific condition, but across the whole map. So that way you can actually see if you had a specific plant, let's say hypothetically one lemna, what would you expect to see? An increase in growth or a decrease as you grow, as you change the level?
Did I hear?
So this is what a lemna looks like. Like, I don't want to give it a cocky look, but try to understand where I'm coming from. This is one lemna. You want to have 3 lemnas looking like this. Right? Sorry, 4 lemnas. So just remember, you want to have them, and they all should be healthy, by the way. Make sure none of them are like pale white or looking like they're about to decay. Because we want to control for their any anomalies that we may or may not be able to expect. Was there a question? No? Okay.
Yeah. Yeah.
Okay, so in the meantime, you could get ready your dissecting microscope. You'll be analyzing the tetrapod, isopod, the sowbug, Porcelio subspecies. Make sure that for your assignments that you put the italics with the name. It also is beneficial to future MLRs.
This is different from us. Oh, yes.
Okay, so hopefully now everything is arranged. Make sure that you place them alphabetically as well, your tubes or your reps into the appropriate location. Now that you're using the dissecting microscope, I just want us to bear in mind time. I want us in the next 5 to 10 minutes, about 10-ish minutes, to be able to look at the isopod, sowbug in the Petri dish of your dissecting microscopes. So each of you, like each group of 4, should be able to visualize it accordingly, or each group of 3 from your microscopes. And then you should be able to look at an example of it in your lab notebook as well. If you want to know the name or the location, have a look at the background in page 62. You'll see a diagram of an isopod, and hopefully you'll come to appreciate the different features that are also shown here. So you have the antennae, as I'm showing up right now. You have the antennae. You have the different features that are also asked upon for you to draw with a figure caption. So note the following pieces: the abdomen, right? As you can see here, the abdomen in this location, this region, also called a pilion of 5 segments. It's near the back end of the sowbug. And then you have the compound eye, which hopefully connects us back to Daphnia, because if you recall, they also have a compound eye. And so you can also see that from our previous week's lab 3. But nonetheless, you have this as well as the head, the legs, the telson, the thorax, and the uropod. And so hopefully you should be able to see the different components, uropods at the back as well. The pleotelson, also similar to the telson, is towards it's actually above, more anterior from the uropods. And then you have, as shown, the thorax between the 2 eyes. So hopefully you should be able to draw and annotate the different features. And if you need, hopefully you all actually have a diagram as well. So you could be able to look at them accordingly and see if you can match the details out in the next 10-ish minutes. Or actually about 5. We'll give you 6 minutes to try to complete this. And if anyone has problems or is unable to see the uropod, let me know. And accordingly do so. Folks, don't just look right away. I want everyone per group to actually look at the insect, the sowbug, because for the next exercise, you'll see them running around. So you won't really be able to take detailed descriptions or annotations of their ultrastructure or their morphological traits. So try to do this now, so that way you complete it for your lab note. And that way you can finish more before the end of the So by the way, insects are very sick. And even like I don't like testing. So that's And folks, please make sure that you're actually drawing it or taking a photo from your phone, but don't just look on like online for like drawing out your pod. That's not the point. You have to live organism right in front of you. You want to be able to take the animation that you do. So bear that in mind. So that way you can actually reflect from reality what you see in terms of their biology. By the way, is there a group that took the glass cylinders here? Is there a group that took the glass cylinders? I noticed that there is a jug, the tubes, the stock, and the artificial pond water, as well as cylinders. That's it. I just want to help the lab techs because they'll come in and they use, so we'll help them out. And in the next 2-ish minutes, try to be done visualizing the test. Next up, make sure annotations label the specific regions that you see from the microscope. So check out the uropod, the thorax, antennae, the biotelsin, so like the telsin, the head, legs, compound eye, etc. Try to do this as soon as possible, so that way the remainder of the practical can be done efficiently. And if you need a bit more time, let me know. But I want us to do this very shortly. Another point that I want to highlight for you. Some of you may have not known this, but for figures, you want to always be detailed in your captions. So let's say this is figure 1. Folks, let's pay attention if you're not visualizing or annotating as of yet. You want to be descriptive with your figure captions. So, for example, visualization of morphological or physical traits or features, traits or features of sowbug. And remember, you put the common name as well as in italics the scientific name, which is Porcelio subspecies, which we have as SPG. If you're writing this handwritten, you can also do an underline for the scientific name, but you want to be able to include both and distinguish both of them, the common name and the scientific name, just as how we did in the zoo, and just as how you would have done for your other like research questions from our previous exercises from the past few weeks. So, and then thereafter, for like an FLR, you would include more details, right? For example, let's say you are actually analyzing specific conditions in your sowbug, then you would include those details, right? And I tell students this because it's very important that whenever I'm marking, that you're able to provide as much detail as possible, such that if I were only to look at your figure, I'd already know what your formal lab report or what your analysis is undertaking. So it could be a graph, it could be a table. Tables are having captions above and figures are normally below the diagram. But you want to also include the conditions, specifying the condition, right? You want to provide as well briefly what your findings or results are. Different labs may also say you don't have to put the findings, but for our purposes, it's better to do so. And then any concluding remarks or interpretations of your data. And this is something that you want to be able to describe, because even from this figure today, as a classical example, you want to be able to see, okay, am I able to find all 7 or more traits
Can I see all 8 traits indeed, like the abdomen, the antennae, the compound eye? Do I see only one eye, 2 eyes, etc.? Does the erythropod look healthy or not? Does it have other unique features that are, outstanding from the classical illustration? So you may see like a larger, like one larger antennae over another, or 2 extra legs, essentially. So these are things that you want to consider. Like, for example, some organisms have, different unique features that you may not always see from the classical descriptions. So as we move further, by the way, everyone's done? I'll move further actually so that way we're able to keep track. This exercise for today, I really want to make sure that you understand what's expected, because you're doing an experiment once again in this exercise. And so if you look at exercise 3, page 67, once you're done, the illustration of the terrestrial isopod, actually just to make it easier, you could also include a terrestrial, which is like a land-born or land habitating terrestrial isopod / sowbug. You can include both in your description. That's something I want you to consider for your figure. Moving to exercise 3, you have so let's look what we have around us and what we're aiming to explore. If you had a look at exercise 3, you're looking at the behavioral responses of isopods, meaning essentially you're going to use each group, by the way, each side across all groups should have 2 separate traits. One that's labeled as control, which is probably along this side, and one that's labeled as experimental on the other side. The same applies for the other end. Both that are on the left side, please make sure you're paying attention, because you'll need to know these instructions for a final portion of our practical together. So you're trying to explore behavioral responses, right? And the goal is that you want to see how different substrate conditions influence the mobility or the response or taxis of your isopods. So, for example, if you have something that attracts an isopod, they'll move that direction, move towards that direction, like chemotaxis, in chemotaxis as an example. Or if they repel, let's say they don't like a specific compound, they'll move to another well or a location. And that's what you're trying to investigate as a group. And essentially, you have different options as shown here. On each bench side, you'll have in one of 3 options in a specific red ice bucket, or in a specific red bucket. That is either chemical substrates, or sugar substrates, or food substrates. And then you have 4 different categories of them shown on the slide, but also on your buckets as well for the experimental tray. So you want to bear in mind that you have 2 different trays, because you're going to have one as a control to see whether or not there indeed is even a relationship or response to specific to essential of the isopods in your control tray. And then you're going to translate that to or apply that to your experimental conditions. And you as a group can choose, but it's often easier to do it on the bench top that you have here. So for example, on both sides, you should have food substrates. On these sides, you should have sugars. And down at this end, you should have chemicals. And so each category or each condition will have 4 different, each category will have 4 different conditions. And that will be well served, because the experimental tray will have 4 outer wells. They're already labeled, right? And the central well is your well 5 or well 0 that you can consider for your control and your experimental trays. But the idea is, now you're going to essentially observe how or where these isopods migrate to, right? And so you want to think about insects, right? Where do they migrate? And what would they likely want to essentially go towards? And so the dependent variable is essentially the number of isopods that you find in a particular well. So bear that in mind. And then the variables that you want to control for, I actually want you guys to think about it. So think about like what we've done for the winter wheat, what you've done for today for the lemnas. Remember that, for example, we controlled for water, the appropriate amount being consistent at 180 mils, except for the control, right? And then for the other conditions, bearing in mind that you use 20 mils of your stock solution, that was also consistent. So you want to consider these controls and other ones external towards our environment to be able to analyze the effects that they have on the cell buds. Now, this is the fun part, and before I move further, any questions? No questions? Okay. So this is where you guys are going to actually open up the vial of your cell bugs. So you're actually going to open them up, and I'm going to explain it to each side, so that way you guys can do it in the next half hour. So let's see from the middle bench. So that way everyone. I should do right on screen to each because I also have to open time. That's another point. So you guys should have 10 bugs. You see 5 here as an example. I actually skipped some slides so you can understand where I'm going. I believe it's this one. Yes. So have a look at your bench top. You should have a paper towel in which there are 5 different filter papers on each side. I'm going to check this. Have a look. Have a look at that paper towel. It has 5 different sets, 2 pairs of filter paper on either side. And the goal is that one's going to be for your control and one's going to be for your experimental trait. So you can start putting them in right now into each of those wells. And the idea is, actually, I'll let you guys do that now. So we'll do it a bit interactively. For each well, you should have 1 filter paper, and from that paper towel, you should have 2 sets of 5 filter papers. So that way you can add 5 into your experimental and 5 to your control. And then the same for other groups. You guys will actually, folks, you'll need to do it for all sides. I know we're working as groups of 3 mainly, but you'll need to have 1 member of your group on another side. So just bear that in mind for doing the other trade. And if you need help or assistance, let me know.
You do water on yours, and we'll do stuff on ours.
Yes, so before you begin, don't place in the substrates as of yet. I want you to bear in mind that your control tray will have each, so look back at your benches. You should have vials, small vials for your control. You should have 2 of them for each block of your group, because 1 will serve for the entirety of your control tray, i.e. you'll be adding in water across all wells for just the control. And then for the middle well of your experimental, that's the only one that you'll be adding water to. So 1 will be enough for control. Actually, but before you move further, folks, this is where it's important to be cognizant. You are adding in water briefly to the periphery of your filter paper. So I'm going to show it on this side, and I'll come to you guys in a moment. In the meantime, try to separate out, like actually make note even in your manual what bacteria, what conditions you're looking at. So I'll put on the So this, you will have 5 different traits. Technically, I'm using this file, but the idea is, most actually come down to that. Yeah. So everyone comes, so that way you guys can come cameras. So the filter paper can be on and you want to be careful because we're just mindful that you're not over adding quantity. So you're just adding it to your periphery, and you want to be sure that the filter paper is soaked, but drop by drop. You may take a bit of time. That's perfectly okay. You want to make sure that you're not over adding water, so that way it's not over humid. Some they like it when it's dark. It's not considered very close to me, but you don't really let's not be your results. So you want to repeat that with this good control on all 5 minutes. For the experimentals, you would do the stages with water, with water. And then just let it not dry off, just let it essentially be stagnant for minutes, but then for remember other wells, they should have labels. Just to cover it to maintain humidity. Always when you're actually dealing with the sowbugs, they actually are inside. Make sure that you represent. Yeah. Something super dynamic for punch. I'm just across now the question is, and then when you're done, cover up the main entire wall tray with the last year to maintain the moisture now let's do no, yeah. I don't know. I think it's like. Yeah. That's another thing. All the way from but like I can eat that too. Like I can see that I got a lot of energy. I don't know, man. These ones, I mean, I'm waiting. Yeah. I really right. Nonetheless, very common points where, you know, what? Oh, you got I just water bad protein for 2 stars. Okay, yeah. No, that's for a different one. That's for theirs. You have. We don't have. They're different experiments. Yours is a tough time doing sugars, right? Yeah, that's fine. The first one should all be water, so they should all be pretty much the same. In the middle section, group one, and then we'll talk about it. Try to take this out. And then we'll go further.
Well, I think it's still pretty weird because they have a lot of.
Any questions, audience? By the way, if your group crosswalk relevant, any help? No. So, folks, what I'm gonna do for the middle batches, please pay attention as well, because I want to make sure that you're understanding what you need to do. For the control, all wells are going to be added into quadrant, for your filter paper. And how you're going to do it is, simple, but, something that's peculiar is that you're making sure that you don't I was going to say, overburden as well. Like you don't want to overuse or over add as much water, more water than you actually need. So have a look, folks. I just want to make sure everyone's paying attention because you want to follow the same for all wells, both for your following the conditions that you have. So you'll add in water, starting off with the edges better. So that way you don't have to overdo the sector. And that is like soap and essentially penetrate other regions slowly. This will give you 1-2 drops at a time. You don't overdo it. And then once the whole wall is covered, repeat the steps for the other three in your control. Right? For your experimental, you're going to do the same, except only the middle walls with water. And then you want to follow through. Actually, when you get all 5 for both sides, you'll cover this up to maintain moisture because sowbugs love when it's moist and they also like when it's dry. So bear that in mind when you're after started for the conditions that they control. Then once you get all 5, you do the same. And then I recommend taking off the pipe tools because either in the sowbugs. That's I think this should be also. The point is, you want to bear in mind the 2 different categories. Category 3, and they have already pipettes labeled. Please make sure you're using the right pipette because, as you can see, each wall corresponds to a specific pipette, and each pipette refers to a specific liquid or compound of interest. So for example, I'm looking at starch. That is wall number 4. I need to take out pipette number 4. It's also labeled as starch. So make sure that you're picking up the right one. The middle wall goes with water once again, and I'm going to mix, make sure that it's good as well. And you can actually also do other practices that you may have in the future. Just bear in mind that when you follow the same exact quotes, you add in to the edges, and you take your time. It's okay if it's some compounds may take a bit more time to loosen up the filter paper. But you want to make sure you're not overdoing it. And if you find that's the case, let it essentially soak up a bit more longer over the direct light. And then moisten it up once you're done, all 5 wells, you can cover it up with a glass cover, and that should be and just bear in mind that you use the right pipettes for the appropriate conditions. Remember, the middle wall for all trees should be just water. Any questions? Any questions? And then so once you're done, cover it up for all 5, and then you take out those glass so that way you can actually get started with So, each side should have, So, yeah, I read So try to do this in the next 3-4 minutes, folks. And then I want you to actually go straight ahead to essentially taking out the sowbugs. I want you to also, for those that are almost done, have a look at the sowbugs themselves. See if they have 10 and if they look healthy. Because if you're having, let's say, 7, you need to let me know so that way we can get more for you. Because that way we want to be able to have a good amount in each well as a control. To account. Some sowbugs, by the way, may not actually be dead. And by the way, my recommendation is that you actually place them on the pipette cover, sorry, the glass. Like try not to use scrupula, use the spoons. Spoons are easier because you don't want to sharp, but you don't want to kill or harm, you don't want to harm them actually. Let alone hurt them. So as you have, my recommendation,
Wait.
so this is a recommendation. Are you guys all verified? All three? Group 1B, are you guys done all your grades? Are you guys done all? Yeah. So this is what I'm gonna ask. You wanna have them close for this side. I want you to pay attention. You're gonna essentially place it in either the glass middle well or onto the dish itself. Then you put it gently press on the other edge. That way they all come out together. Folks, please make sure that they don't come out onto your benches. Make sure that they stay onto the trees themselves.
Yeah.
No, that's a bug. Scroll down. Yeah.
Oh.
No.
Oh, I see.
Yeah, no, I just literally. That's not what we expected. Here, you look. Oh, you got everything? Yeah, you do that. You got it. I just I'll cover it right when you do it. So you have 2 minutes that we got. We have. Great. How did you play without the machine?
Let me know.
Okay.
I think they said that was how we do them.
Yeah, well, that's a bit of a problem. No, we're good. Okay. Watch this. I'm going to scare them away from this side. Then I'm going to focus. Then it's super good. Watch this. There we go. Here we go. What do you do? This, okay, the number is easy. Whoa. Whoa. Yeah, no. Why? Yeah.
Please pay attention if you are missing sowbugs. If you're missing sowbugs and have some other guests, please let me know. I'll work that. Yeah, everything's okay now. All 10 sowbugs, right? All 3 groups. For both times.
Yes.
Okay.
So in like 3 minutes or 5 minutes. Yeah. You have 11. You go out. Okay. Yeah, I don't know like I do. Can we take one? Oh, no. Just wait 5 minutes and then we take off this and then just measure where they are every minute. Let's do okay, yeah, one and everybody.
Incubation.
I don't think that was
Not needs to be on top. Please make sure that you're controlling your humidity in that way.
Yeah, I would be like that active. Well, we do our that. I don't think I can do it. That's the one. We can do ours anyway.
This is collaboration as well. It's really
Well, I need you to tell me how many more back, solvents you need, so that way I can directly ask the last next, community. So I'll try to get each one.
Okay.
We have.
What? No, we don't need water. But we have. Yeah. Well, you want to start? This for the year when it was.
First one,
Yeah. Sure. You can do it for both of us. Can you make it so it just goes off every 1 minute? Yeah. I mean,
Yeah.
yeah, already. I really, a little bit surprised. Let's see. It's like repetitive. Right, you remove this up back on. Wait, what? Yeah, yeah. Yeah. One gets three is one after. I'm waiting. That's from that one. This is cool. Okay. Oh, okay. Zero. Yeah. This one is zero. This one is zero. I did it. Easy. Three, four.
Mohammed. Mohammed, are you here to do this? Yeah.
Okay.
I did it, man. Oh.
So, I haven't mentioned it as of yet, but I just want to let you know that as you go forward, folks, let's pay attention. If you aren't aware, your next steps are now recording the number of sowbugs you find in each well. There is a table that you will see this for the control trial, the control trays. That's page 69. And page 71, you will see it for the experimental tray. So folks, I hope you are recording the values that you're seeing of the wells. If you haven't done so, do so now, starting at 15 minutes. Does every group have 10 sow bugs in each tray? Yes or no? 10? Okay. Remember, folks, as you're counting, the glass tray should still be left on. Only when you're taking out the sow bugs at the beginning and at the end is where you're actually thereafter taking out the middle petri dish. Apart from that, the glass tray should always be on top. And hopefully you guys are recording the values. As you're going forward, I need to make sure that you're all placing the trays, the glass trays on top, and you're appropriately recording the data. One group will be recording, both. Attention, attention. We're paying attention. Aaron? Okay, let's pay attention, folks. As you're recording every minute, you need to make sure that you're keeping track of the lab manual. You have pages 69 for the control, pages 71 for experimental trays. You need to be recording every minute for the next 15 minutes. The location or the number of sowbugs in each well. The middle is your center well, and the well 1 is on the top left to the top bottom left for well 2, and then it goes up to 3 and 4. You just want to make sure that you're recording the data accurately every minute, and you're able to thereafter determine further analysis in the next 15 minutes for your last time point. I will give you until 12:10 to be done. That should be enough for 15 minutes. If you're done as well, exercise 1, looking at the cell bugs, you should shut off the dissecting microscope so that way it's not irritating the cell bugs. Remember, they grow in dark, moist conditions. So shut off whenever you're done observing the cell bugs, to shut off the dissecting microscope. And use these conditions as key points for your post-lab assignment for lab 5. Are you guys?
Because they swap like last second.
Yes, so at the center wall, there should be 10 at times 0. I recommend, by the way, if there's a major interruption, you can pause, but don't generally always pause every minute. You should try to record seeing visually the someone and the number of each well, and that should take about 10 seconds. So you can do that when it's 55 up to 0.5. We count the community that
Like they all just start moving right when they know like they're chill. Yeah.
Yeah, so you may actually see some of them interacting with each other. Some of them are like playing around, some of them are rolling around, some of them are like just quiet by themselves, right? So you can appreciate the different personalities that isopods have. They connect or translate these behaviors, what you saw them do. Like the Arctic wolves that like to migrate with each other, versus certain species that like to stay alone, or jellyfishes that like to flotate in different unique arrangements. So you want to see and appreciate these different wonders of life, including the isopods in front of you. When you're done, I'll talk about that now, actually. What am I gonna do? I can't do anything. That would be nice. I don't want to tell you. Yeah. Actually, I wouldn't do that. I hope at the very end of all practicals they let them to the wild. But when it comes to completing the lab, or at least completing this, I'll mention it at the very end for everyone. But can I just get a rundown? How many of you need 10 minutes? 10 minutes? Yep. How many of you need more than 10 minutes? Raise your hand. Okay. Okay. So let's see if we could be done in about 10-ish minutes. And as you see these isopods move in live action, I want you to write down underneath the tables what you're observing. Do you see them migrate to a specific well or not? Do you see them go to multiple wells or stay in one well? Do you see them interacting with each other, trying to, obtain the resource from the filter paper? Because for them, they may think of
the sugars that you see or the food or chemicals as things that they can actually absorb, right? So think, or like to intake accordingly. So think about those different patterns and what you're seeing. And just bear in mind that you're paying attention during this every minute for the or regularly as you're observing the time.
So that's, you can just write it up there. I don't know. You have an extra one.
So think about your observations, record them, and think about what they mean. Interpret your results based on the different categories and different conditions you have.
Yeah, I think you lost. Okay. Yeah.
You have taken the wrong lid. Has any group taken this lid accidentally? Lemna lid? Has any group taken the lemna lid? And they notice this one was placed.
Yeah.
Because that one was lemna.
Can you do one? Can you add up all the columns for me?
How's the movie?
Yeah.
So once groups are done, what you're gonna do is actually take the groups. We're essentially gonna bring them back into here. And I can help you then. The other thing is, you take the spoon, you place them into actually here. Bear in mind, you're gonna place them into here first because you want them to dry off. I'll talk about it for the group to come. Just make sure that you dry them off. Let them dry for 3 minutes. 3 minutes, and then you essentially and then thereafter, you can use the paper towel and gently burn them to all of them down.
You're right.
Moment for one group. Yeah. And yes, you guys can swap data and you can take photos because it will make it easier for you to record for the postdoc. Yes. By the way, don't use the metal, don't use the sputulas. So that way you don't run the risk of killing them. Use the spoons to take them into and you have many spoons, hopefully. When you go
Wait,
So for context, folks, you guys are 45 minutes or about 40 minutes early. So try to see as you're doing what you're observing from the isopods. What do they favor? What do they repel from? These are traits that you want to analyze and be able to appreciate 50 minutes into your exercise into the lab or incubation. So let them join on the tissue paper, the paper towel for a few minutes, and don't let them go out to other locations. What's it? I just want to open the first. Yeah. Yeah, okay, no, I don't want to show you the. Yeah,
Why is turn these guys on? Wait, what did you do? Oh, you doing
By the way, it's like this. Remember, also brought back into let them paper towel, let them dry off for a few minutes, and then you just place them back in. That way, the water that was augmented with the liquid from your experiment is essentially precipitated, and then you don't get it.
expected number here? For the stats, the expected number of animals, is that like you decided? Yeah.
Yeah.
So when your group is done, make sure that you're putting down the trays, don't stick it out, and then you should be able to thereafter throw the clean material, wash your hands, and throw the filter paper into the garbage. And then return back to center plate.
Steve, like, okay, let's start with the here. Alright. What, yeah.
And folks, remember that you gotta put them into the paper towels, before you put them back into the vials, so that way they can dry off their antigen, their lettuce accordingly. So that way the future products. And then when you're done, you can take the paper out, wash your hands. Return things back to where they were, the milk tray, down to, the petri dish down to the well, and then the cover onto the top of the refrigerator. And then that will cover, then we'll cover the back. Okay. So we'll wait another few minutes, about 4 minutes, to make sure everyone is done. When your group is done, please let me know. Putting back the isopod.
Yeah. No. Use. Oh, yeah.
They don't give you guys a lot of that's why you're like, sure, one is a one.
I mean, pretty bad. Or someone had to. Yeah. So then. Oh, actually, no. Oh. No, I don't know how to do it.
Now, you guys are done. Let me know. By now, your tray should be empty with the glass tray on top and the middle well with a petri dish, like the one before. So just make sure you put it inside. Inside. Either way, inside or on top is okay actually. But I just want to make sure everyone's okay. So I just want to confirm, Mandarin, what the amount of files, we don't, and I can pass back to, all of them are healthy, but, yeah. I want to make sure you take other labs.
Yeah. And I think it's.
Is it wash your hands? We'll talk about that. How was that for your second math lab? Good? How does everyone feel? Everything got for you guys feel okay? You guys feel good? You guys feel okay? You guys feel like it was good? Yeah? Did you feel like you got to touch some isopods in real time? Everyone felt good? Bishop felt good? Yeah? Yeah? Wanyan was good as well? Yeah? Okay. So let's talk a bit more as to what you expect and what you need to take into account for your statistical data analysis. Now, unlike our previous labs, you're dealing with a chi-squared analysis or chi-squared test. And as you can see from page 72 and from page 70, you'll be able to record the different values that you find for time point 15. So your last time point where you saw like the isopods at their final moment in their wells or in their respective wells, you're going to use those values as your observed numbers for each category. And you know the categories based on what is shown in the ice bucket, right? So number one for sugars is glucose, then it's fructose, then it's maltose, then it's galactose for 3 and 4. You want to be able to record those data points, and then the center well being water for all. And then similarly account for those details for the control, which will all be water. You'll also want to be able to highlight what your null hypothesis and what your alternative hypothesis includes. Your null hypothesis assumes that there's no significant difference between your observed data and your expected data. Your observed data is once again the number observed is essentially the number of sowbugs at t equals 15 minutes. Expected is what I want you to do the math for. You had 10 isobugs, right? Who had more or less? Please let me know now. Anyone had less, more than 10 isobugs, solvents? Okay, and then 5 wells. So if you do the math, how many should you expect then in one well? 2, right? So bear that in mind. That's your expected number. You're going to be comparing essentially across each well what you would have expected per category, right? So, and comparing that with your observed values. So you're essentially placing in your categories, as we know, your observed values, as I talked about, and then your expected number, which is 2. And then you're doing the formula that you could do either on the calculator here, you could do it on your phone, and you could do it on your computer as well. And then hopefully you'll amalgamate or accumulate all those values from the last column of table 5-5 and table 5-3. And you'll like add up all those values for the last column into the last bottom right cell or box. And that's what your chi-squared calculated value is. It is essentially the sum of the difference between your observed and expected values squared divided by the expected value itself. So essentially it's your observed value divided minus 2 squared divided by 2. And then hopefully adding up those values per category from your observed values per category, you will determine your chi-squared calculated value. Now, you have another value as your critical. There's also a short form for this, but the idea is that I don't know if it shows you here. We'll talk about it in due time, but you already know that, like, for example, a p-value that is less than 0.05 or equal to it would reject your null hypothesis, and therefore you would assume that your alternative hypothesis is supported, or that, and hopefully we're, let's focus, folks, actually, it's very important. The p-value being equal to or less than 0.05, you're rejecting the null hypothesis, and whereas if it's greater than 0.05, then you fail to reject the null hypothesis, i.e. that your observed and your expected values are assumed very similar. Similar enough, indeed. Whereas if it's rejected, your null hypothesis, then you assume that there is a significant, or there is a difference between your observed and expected values of where your isopods would be located based on their specific behaviors. And so the point of this that you may want to consider is that the degrees of freedom is 4, because you have 5 categories, 5 wells for each trait. Subtract that by 1, and that's 4. Now, you'll use the degrees of freedom and the p-value at 0.05 to determine what your chi-critical value, chi-squared critical value is. Can anyone tell me what the critical value is right now? You can also see this on the book, so don't worry about quotes, but I want you to look and tell me what values you see would give you 0.05 of the significance reading with a degrees of freedom at 4. What is the chi-critical value? It's essentially where the two overlap, where df equals 4 and 0.05 is your p-value. Where do they overlap as your critical chi-square value?
9.488.
That's one option. Any other, or that's one response. Anyone else having the same response or different responses? Raise your hand if you have a similar response, like if you think it's 9.488. Okay. For those that don't think so, I want you to tell me what other values you're looking at. What other options do you see? So the correct answer actually is 9.488. I just want to let you know, and just to see that if you guys are aware, that it's similar to thereafter your results in comparison. If you find that, for example, actually we know now it's 9.488. So based on your calculations from table 5-3 and table 5-5 for the control and experimental tree chi-squared values, if you find them to, once you find out the result, you want to be able to compare. If you find that your calculated chi-squared value is greater than the critical or equal to it, then you reject the null hypothesis, right? Similar to a p-value that is less than 0.05 or equal to it. If though you find that, and that's if your chi-squared value is, the calculated one is equal to or greater than the critical. So even if it's a 9.489, right, then that rejects the null hypothesis technically. If it's less than it, like 9.487 or less than that, then you fail to reject the null hypothesis, and therefore you assume that the observed and expected values are similar across your conditions, or at least in terms of the response of what you would have expected. Sorry, let me just write that down to make it easier. So if you have it greater than, just have a look. If it's equal to or greater than, you reject H0, HO, that's the null hypothesis. Therefore, your observed values and expected are not equal. Expected, by the way, is 2 sowbugs per well. If it's less than if the critical value is greater, or i.e. the calculated value is less than your critical, then you fail to reject, and you essentially assume the null hypothesis. And for your lab 4 assignment, please make sure that you refer to both. I prefer you referring to both because it highlights what you know from your experiment, not simply what you don't know. Because if, let's say, the null hypothesis is correct, you assume that
indeed there's no significant difference, but I want you to also highlight that that means that you accordingly would cannot discern or you cannot support your alternative hypothesis. And so, just for contrast, that's what we're talking about, right? And these results are similar to a p-value that is equal to or less than 0.05, or p-value that is greater than 0.05, respectively, for where they reject or accept the null hypothesis. That's pretty much it. Lab 5 assignment. Please make sure that you talk about in detail or follow through with what we've talked about before for your research hypothesis, your null and alternative hypotheses, the predictions for each trial. You analyze your data from tables 5-3 and tables 5-5 from pages 70, pages 72, and make sure you fill in the details that I talked about earlier for your wells, the setup for page 72, and for 70 and 72, your null hypothesis, alternative hypothesis, which you'll include for your lab 5 assignment, and then your final value for chi-squared calculated in your manual for both trials of the control and experimental trays, but also in the lab 4 assignment, lab 5 assignment, and you'll summarize your data, talking about the trends that you see and answering any discussion questions that I've talked about and what you could consider as limitations from your results, plus what you find out from or what you're able to discern. You can also see, by the way, questions highlighted in page 73. So have a look at them so that way you can prepare yourself for the lab 5 assignment. And that's exercise 4, which concludes our lab as we finished exercise 5 at the beginning. Make sure once again that you've cleaned up sowbugs, clean up your benches as well. Make sure there's no debris. If you're done, then we're done as a group. I congratulate you. We have one more lab and then one more lab 2 weeks from now. And I hope you guys have an amazing rest of your semester. And for those that are like fasting as well, like during the month of Ramadan, I hope you have an amazing Eid, because that's when we'll likely have Eid there before. So folks, I just want to say, once again, really appreciate this time with you. And I hope you have an amazing time throughout the semester. If you have any questions, try to reach out to me earlier than later. You likely have my email as well. And I just want to mention once again, try to be observant and consider what I've talked about thus far. The controls, conditions, discussion questions, integrating different concepts, and like behaviors of isopods, like how we saw behaviors of animals and other organisms at the zoo. So think about these different concepts as we approach our final set of labs. So with that, any final questions before you leave? I'll let you guys, before you leave, I'll just have a look to make sure everyone is also in attendance. If you think that you weren't marked properly in from the past, let me know. Let me know if you weren't. Well, I don't want to show everything. If you, for whatever reason, actually, I'm going to say, try to keep the files like this, stationary, so that way the isopods are not directly in sunlight, and that way they can actually be more shaded. Make sure everything is clean. Apart from that, you're ready to go. Yeah.
Same thing though, but like just doing it at 15, because like look at 15 compared to like something like 7. Like it just seems so random. Like you know what I mean? Like there's like if I chose any of these, I'd get a totally different answer. There's nothing like making one. Okay. Yeah.
One time point before, where essentially just before you got you notice that, like, for example, on all one, there's still
There's similarities, yeah, but like I don't know.
like you could see probably the ones that represent the well one straight well,
Yeah.
right? So you could kind of get back. But I would prefer you
Now you don't win. Now you're not even close to winning.
from the categories of
Yeah. What you said does make sense. Look, like this is like a pyramid. Like it goes up, they're all here. They take all the two, and then one really goes down. So just for the O's, just do the two.
for the for all of them. All of you. Okay. Is in pretty.
Some.
So everything goes good. You got it. That was you.
No, and then they need them to check, and then you do it. Well, I guess. Maybe the septic. Yeah, I guess, because how they, what they're. She's like, I don't get it.
Think about And that's an important reminder, folks, specify the condition. For your hypothesis. Specify the
Yeah, I was gonna ask, like, if the hypothesis you mentioned, like, instead of saying there's no difference. Or there's no difference in the observed and expected convection, like, specifically what the observed.
like observed and expected like if it's four categories, like if it's for the control, it's just their behavior of the isopods, but then must determine what condition are they with the control behavior. Moved that they here, but then for the experimental is also. So you want to specify for the different ones, the different hypotheses related to the control trait versus to the experimental trait. What conditions were involved? What did you add into those traits? What's the filter? Does that help?
Yeah.
Yes.
Usually go for things that's smaller to it, but that has.
Mhm.
Yeah, so what is it? You said you just did it. What, because if you look at these numbers compared to these numbers,
Yeah.
what if they kind of, they went to the other ones at the end?
That's possible, but I see like the fat is more abundant, right?
Yeah.
So that's something like the idea is that yes, it may appear that this is smaller and the proteins or even the fruit juice is more attractive,
Yeah.
but mainly you want to look at like you can do both. If you want to do like the mean, just let me know, specify in the lab assignment,
Okay.
and I won't take it out because it is a good point actually. But at the same time, what they do want you to see is that 15 minutes because you not actually see clear changes because
Yeah.
technically you divide this by 15, right?
Yeah.
So this would become like 3 point something. This would become less than 2, even though your final result is at 5. You see what I mean? So the the mean itself will not account for variation directly. Or like there's still variation that may be subtracted or deducted from the final mean value that you get. So that's why it's beneficial to see the time points in that way. So that way the results are able to be directly correlated to the final time point you analyze.
Yeah,
No problem. Any questions?
that's all. Thank you.
No problem. Have a great time. I wish you all the best.
Yeah, thank you.
Thank you. No problem. Okay. Good job. Yeah. This is the next one.